Hello everyone. Today we're going to talk about protein synthesis. We've been talking about DNA, and today you're going to see the link on how we get from DNA to eventually creating a protein. So first, let's remind ourselves what a gene is. We know chromosomes are in the nucleus. All right, so here we have chromosomes from the nucleus. All of it's made of DNA. And remember, a gene is just a section of that DNA that has the directions for making a certain protein. And that protein will determine your trait. So we're going to talk today about how these sections or segments of DNA with A's and T's, C's and G's end up making traits like eye color, hair color, and so on. So what do proteins have to do with traits then? Proteins essentially are traits. So you might have a gene that has the code for an enzyme, and that enzyme makes the pigment that makes the color of a flower. Your eyes are the color they are because of a protein. Your blood type is the type that it is because of a protein. Your hair is the color that it is because of a protein. Almost everything in your body is either made by a protein or is made of protein. So proteins have everything to do with traits. They basically are your traits. And remember that the directions for making those proteins are in your DNA and genes. I want you to consider this. This is a puzzle that you might have seen, in elementary school. Little kids like to do puzzles like these. And if I had asked you to take a look at this and solve this puzzle, you might have had a hard time. And if you think about it, you could probably solve this puzzle if you had a key that told you what all those symbols represent. This is how DNA is. Right now, to you, DNA is just a bunch of letters that don't mean anything. A's and T's, C's and G's. And it doesn't make any sense until somebody cracked the code. Because to our body, those letters do mean something. So somebody, a scientist, had to figure out what that code means to our body. And once they figured that out, we can solve the puzzle too. So now that the code has been broken and someone has figured out the code, DNA is much easier to read. Just like this puzzle is easier to solve once we know what all the codes are for. DNA is much the same. As we go through today, you're going to be learning how scientists read the code. And there's a few steps to it, how we get from DNA to eventually making a protein. The process of going from DNA to protein is called protein synthesis. Remember, synthesis just means to make something. Protein synthesis can be broken down into two major steps: transcription and translation. Transcription is where a copy of the DNA is made from the directions in the DNA. Remember, DNA is inside the nucleus. The DNA is too big to come out, so we make a copy of the directions that we need, and we send it to the ribosome. Why would we send it to the ribosome? If you recall, the job of the ribosome is to make proteins. So ribosomes don't know how to make the protein if they don't have a set of directions. So once we copy the directions we need from the DNA, we send it to the ribosome. And then part 2 is where your ribosome reads the directions and makes the protein. That's called translation. We're going to go through these step by step. We'll start with transcription and how it works, and then we'll talk about translation and how it works. And then later, you'll put it all together, and I'll give you just a sequence of DNA, and then you'll be able to tell me the protein that gets made. Here we go. Transcription, the job of transcription is to get a copy of the directions for making a protein from the nucleus to the ribosome. It happens in 3 steps. So keep in mind as we go through these, we said protein synthesis is the overall process. It has 2 steps, transcription and translation. Right now we're talking about transcription, and transcription itself has 3 steps, so it's important that you keep this organized in your notes. So transcription happens in 3 steps. First, the DNA uncoils at the gene needed for the particular protein. Okay, so we've seen this before. DNA uncoiled when we did replication. The difference here is we're not copying the whole DNA. Here, we only want to get to some gene, some specific thing that we need to make. So we find that gene and just unwrap the DNA at that particular point. All right, so the DNA is going to uncoil at the specific gene needed for whatever it is that we need to make. The directions from the DNA will be transcribed. Transcribe basically means to copy into messenger RNA. All right, messenger RNA, you should remember from yesterday when you learned about RNA. Remember, RNA doesn't contain any T's. It has U's instead. This is important when you're looking at a question on a test or quiz or looking at something out of a book or online. If you need to know what you're looking at, look at the letters, whether it's DNA or RNA. If there are T's in it, it's DNA. If there are U's in it, it's RNA. DNA coils back up after it's made a copy. All right, remember too that the copy is not an exact copy. A's don't go with A's, C's don't go with C's. So it's a complementary copy gets made. And anywhere you would have had a T, you're going to put a U instead. We'll practice this later on, so if that's confusing, just hold tight. Once the copy is made, DNA will wrap back up, and then the messenger RNA can travel out of the nucleus and to the ribosome. I encourage you at this time to type this website into your browser, pause the video, and you can watch what this would look like in your cells. Of course, remember that your cells don't have color. This is a computer-made graphic, but it shows you how this would happen inside your cells in real time. So it would show you how fast this happens in your cells at any given point in time. Feel free to stop this video at this time and put that website in and watch. We're now going to practice transcribing. So eventually you're going to put transcription and translation together. We're going to do just the first step together. So what I'd like you to do is in your notebook, copy down this section of DNA. How do I know this is DNA? Because it has T's in it. You are going to transcribe. That means follow the steps that we just talked about for transcription. Okay, we're going to pretend that this is the gene that we need. So DNA has already uncoiled and unwrapped at this particular point, and now we need to make the copy into messenger RNA. In your notebook, I would like for you to try to do this on your own. I want you to think about what would match up with a T, what would match up with this T, what would match up with this A, and so on. Make the complementary copy. And do that now. Okay, at this point, you should have the copy written in your notebook how you think it should be. Let's see how you did. So if this was the DNA strand, my question to you is which one of these is most like what you wrote in your notebook? I'll give you a second to take a look. The answer is 4 here. T goes with A, T goes with A. A here would normally go with T, but because messenger RNA doesn't have any T's, I have to put a U instead, and so on. So as you continue, this should have been what you came up with. If you didn't, make yourself a note how to do it and where you made a mistake so that hopefully you won't make that same mistake again. That is how you do transcription. That is the first half of protein synthesis. This code now can go to the ribosome to be translated. Let's find out how translation occurs. Now that the directions have gotten to the ribosome, your cell has to read it. It's kind of like a grocery list. All right, we need this, we need this, we need that, so that your body knows what goes into the protein. But the directions are in a different language. They're in the DNA code, right? They're in, RNA. That doesn't tell me anything. Your body has to read the code. It has to be translated. So it took scientists quite a long time to figure out how your body was reading the A's, the U's, the C's, and the G's. Watch carefully what your body does. When mRNA arrives at the ribosome translates the messenger RNA to figure out what pieces it needs to put the protein together. Your body reads messenger RNA in 3-letter chunks at a time. Not 1 letter at a time, not 2 letters, not 5 letters at a time, but 3-letter chunks at a time. Those 3-letter chunks are called codons. Each 3-letter codon is the directions for a certain amino acid. Scientists have now cracked those 3-letter codes, so we know what every 3-letter code is referencing to tell your body what amino acid to go get. Once we read the 3-letter codon, tRNA goes and gets the right amino acid and brings it back. And then the ribosome hooks all the amino acids together one by one to make the protein. Again, I encourage you to take a break here and watch how this happens inside your body. This is another video of translation, not transcription this time. All right, remember the video of transcription shows just how the copy is made of your DNA, and then that copy comes to the ribosome. This video picks up with the messenger RNA coming to the ribosome, and then what the ribosome does, reading the 3-letter pieces at a time, or 3-letter chunks at a time. You're going to look for the video that says Translation Basic. Okay, so at this time, what I want you to think about is the fact that scientists have figured out what every 3-letter code, every 3-letter codon is the code for. There are various different charts to show it. This is a circular chart. In class, I will also show you a box chart so that no matter what kind of chart you're given on a test or quiz, you would be able to read it and figure out what the codon is for. You don't have to memorize any codons. That's not necessary. We always have a chart to go from. So let me just point out to you how you read the chart. Let's say in the gene, there is a code that says, let's say GCC. What you do is you find in this kind of chart the first letter in the inner ring. Oops, let's go back. So in this inner ring, you would find the first letter, right, G. All right, I think I said GCC. We're going to go with that. So first letter in this ring, so I find G. Now, once I'm in this first letter, I have to stay in this quadrant. So from here, I look for the second G, the C, GCC I said, in the second ring. So I go from G to the C that's in this quadrant. And then I find the other C, and that is the code for alanine. So G to C to C is alanine. What I'd like you to do is to try to see if you can use this chart. Remember, first letter in the inner ring, second letter in the second ring, third letter in the outer ring, and you follow that out to find what it is the code for. I'd like you to pause the video now and try those on your own. Practice is the only way you're going to know if you know how to do it or not. So this is your chance to try 4 now. Okay, at this point you should have written these in your notebook and tried them on your own. Let's go ahead and check and see how you did. So AUG, which one is the proper amino acid that AUG is the code for? Did you say methionine? If you did, methionine is correct. I also should point out here, AUG also has another purpose. AUG is what we call a start codon, or the start codon. It's the only one. So a question you might have or should have is, how does your body know in all these letters where it's supposed to, where one gene starts and another one ends? Well, what your body has are start codons that tell you, or tell your body, where to start. And so every protein, the code for every protein, starts with an AUG. Next, we had GGG. Which of these is the correct code for GGG? Hopefully you said glycine. Glycine is the correct answer. How about CAU? Histidine is the correct amino acid that CAU is the code for. How about UGA? UGA is the code for stop. Stop, obviously, is not an amino acid. Stop is just like what you might think. This is the code, or one of the codes, that tells your body, okay, this is where this gene ends. This is where we stop building this protein. It's done. So AUG is where all proteins start. And there are a couple of different stop codons that tell your body to stop. The protein is done. At this time, you're going to get a practice problem where I'm going to give you DNA. You will do the transcription and you will do the translation. So see Mrs. Smith for that practice sheet. Good luck.